The title of this presentation is Prediction of Genes from Prokaryotic Genomes with GeneMarkS. So to begin with a presentation overview, I'll first be discussing a reference for GeneMarkS with a description of the workflow that the different algorithms use for processing data from genomes, as well as describing and outlining the command line procedures and interfaces that are used for compiling the algorithms for enhancing our prediction capabilities of genes. And we'll also discuss results from one iteration of GeneMarkS with a sample FASTA file that I fed to determine what would be the different types of predictions that the online algorithms would output. So this is first a reference from 2001 from people at Georgia Tech, in which they describe GeneMarkS, which is a self-training, unsupervised method for predicting genes, starts in microbial genomes. And there are also other implications that they list for being able to detect and predict different types of motifs or gene sequences in regulatory regions. In one of the figures that they provide in this 2001 reference, they describe a couple of different steps for processing the data. First of all, in the upper part of the figure, they describe through the initial steps, step one, in which we're looking to build a heuristic model for determining the relevant parameters, as well as two, finding initially sparse sequence and identifying coding regions with a hidden Markov model that they list as gmark.hmm, as well as in the third step, which is the final initial step for the preprocessing of the algorithm, which involves the RBS model building. So we're looking to make use of initializing some parameters that can help us determine different types of sequences of interest. And then from this, after having some DNA as input, being able to make some useful predictions. Besides these three initial steps that they list, the main cycle also consists of steps 4 and 5, which they give as a prediction step in step 4, and then as step 5 lists a model building step. So there's very much an iterative process between steps 4 and 5, which depends on the convergence check that they provide. So the possible output formats that are included for GeneMarkS rely on either FASTA output for the length or the possible numbers of each gene, along with the sequence itself, as well as some FNN output, which is generated from the hidden Markov model, which contains the DNA sequence, as well as for the third possible output, FAA output, which consists of the genes numbered in the order in which they are predicted, as well as one could expect a PDF of all the results compiled above. So we're going to discuss how, depending on the FASTA output, which includes different DNA sequences that can be input into GeneMarkS, what would be the type of predictions and how would the predictions for additional genes from the specific type of genome that we discussed, how would they relate to characteristics of the genes genetic sequences themselves that are used as input for the unsupervised model. So to give one example and to further discuss what GeneMarkS is capable of outputting, we can see that for the FNN output format, we can see that each one of the sequences from the FASTA file is initially parsed. And what it does is that it looks through however countably many sequences there are, and then it makes a decision about what would be the length of the predicted genes from the input length of this sequence, as well as looking at different ways to predict various classes and other classification metrics for different sequences. To go into more detail about different ways that the genetic sequences could be classified, we can see that starting from the rightmost column, we have the class in which all of the potential genes are listed from 46 to 52 that the algorithm predicts for this given FASTA input sequence. They all belong to class 1. In the nextmost column to the left, we can see that for the gene length, there is a variety of different parameters and classifications that the algorithm can make depending upon the intron length or the exon length, which can help it determine what the total base pair length of the genetic sequence is, as well as in the next two columns to the left, the right end and the left ends, as well as the overall gene number from the overall list of genes that are combinatorially predicted. And to provide another example of FNN output format, we can see that in the first example, as with the first example that we gave, there's some FASTA file from the output that's parsed initially from the input FASTA file that were generated from some transcriptome assembly procedures. And then in this case, we can see that there are different regimes and ranges of the parameters beyond which the algorithm is not able to perform as well or form as sharp of a classification as it can for other sequences, in which case, when there's a certain length or limit to the right end base pair length strand, we can see that as given in the second genetic target that's predicted from 3260, that the prediction algorithm can lose some specificity in the end of the right strand that it predicts and instead prescribes a range of possible lengths depending upon where this right parameter can end. So, and then one can see that in comparison also to the previous example that was given, two of the first genes belong in the prediction class 2, and the last one belongs to the prediction class 1. So there can be some examples depending upon the types of motifs and certain specifics of each type of sequences and whether there's a conserved region or other features or characteristics of the genetic sequences that the algorithm can form different types of predictions on from different possibilities that these genetic sequences could hold. But besides the previous two examples, some sequences can also be on the order of thousands of base pairs long. We can see that in this case, for another target that's read from the FASTA file input, that for gene number 3488, the predicted gene length can be almost 3,200 base pairs long. And besides these, the output from the FNN file format that was given, we can also see that there's an output from a hidden Markov model in which the gene output, it differs a little bit, but this is one of the intermediate file processes that, or, one of the intermediate processes in files that the GeneMarkS algorithm generates for forming the prediction for the genetic algorithms, or for the gene sequences from these algorithms. And one can see just that this is a pretty typical sequence length that's predicted from all of the genes that are given in the FAA or in the FNN output file formats. And we can also see that as a longer example, the output from the hidden Markov model can also generate some more longer, more complicated genetic sequences. And so for a conclusion, we described GeneMarkS and we motivated applications of gene prediction. We were primarily interested in looking at how this would behave for the specific type of genomes that are listed and described in the 2001 reference that I provided at the beginning of the slide, in which the authors of that study were interested in looking at enhancing prediction for different types of genomes and how they made their resource and their algorithms publicly available and even able for us to run online so that we can check what the different type of outputs would be. From the possible output files that are given that the algorithm produces at each step from the workflow that we also described earlier in the presentation, we provided some examples of the output, including examples of the FAA output, as well as examples of the FNN output. And this is interesting for at least for future potential applications of prediction pipelines, which remain. And thanks for watching.